Good morning, everyone. I'm Suli, and I supervisor no amongst that. And the topic I want to talk today is the confocal microscope image of the passive transport of carboxylic acid across lipid membrane. And the passive transport play an important role in human health. It's including the inhale of gases through the lung epithelial cells, and the uptake of drugs and drug carriers into the intestinal cells, and the penetration of environmental toxins. So to understanding the mechanism of the passive transport can help us design useful drugs and avoid dangerous toxins. And what kind of factors will affect passive transport? Over 100 years ago, Overton proposed that we can consider the lipid membrane, the cell membrane, as a hydrocarbon barrier because the cell membrane, in the middle of the cell membrane was composed of the isotyl of the lipid molecule. So if the drug molecule has a high oil solubility, it will have a high permeability through the cell membrane. And it can also be expressed in this equation. The membrane permeability is equal to the oil-water partition coefficient times the diffusivity inside the membrane or the thickness of the membrane. And here, the membrane permeability is proportional to the oil-water partition coefficient, as shown in this figure. But recent research about the passive transport of carboxylic acid through lipid membrane has shown a result contrary to Overton's rule. Here, Grant and co-workers found that as the isotyl length of carboxylic acid increase, their permeability decrease, which is totally opposite to Overton's rule. And Xiang and co-workers found that as the isotyl length increase, the acid permeability first decrease and then increase. So they suggest that the Overton's rule need to be revised, and some other factors like the molecular size and structure need to be taken into consideration. And besides their research, let's see some other researchers' results. As you can see in this table, the other researchers' result for the permeation of the carboxylic acid, there exists a lot of a huge variance. Some results obey Overton's rule, and some results violate Overton's rule. And even for the same acid, their permeabilities varied a lot. So this means that there must exist some flaws in their experimental operations. And here, planar lipid membrane and the liposome are the two mostly used systems to test the small drugs permeation through the lipid membrane. And for the planar lipid membrane, the residual solvent existing in the membrane will greatly affect its properties and its stabilities. Make this planar lipid membrane properties unlike the true cell membrane. And for the liposome, it's a solvent-free lipid membrane, which has the properties more like the true cell membrane. But as you can see, the polydispersity of the liposome will also bring artifacts to the permeation results. So a more accurate method needs to be developed to have a clear understanding of the mechanism of passive transport. And here in my system, I use the giant unilamellar radical, which is also called GOV, to compose my model lipid membrane. And this lipid membrane was composed of three kinds of lipid generated by the electroformation method. And the confocal microscope image shows a GOV with a diameter around 6 micrometers. And to track the transport of carboxylic acid, a pH-sensitive dye, fluorescein dextrin, was encapsulated inside the GOV. And trying to observe a single GOV, this GOV was immobilized on the glass surface by the adenine melting interaction. And this GOV was immobilized in the microfluid channel. This channel was composed by bonding the PDMS channel to number 1 cover slip. And this device allows us to exchange the buffer system quickly and uniformly. And the spinning disk confocal microscope was used to observe the single GOV. And this technique allowed us to observe the entire permeation field in a very fast speed. And as you can see, the below figure is the GOV immobilized on the glass surface. And figure B is the GOV's profile shot from top, left, and front. And here are 2 videos. Shows how the fluorescence intensity change inside the GOV when the acetic acid permeates into and permeates out of the GOV. As you can see, as that acetic acid permeates out of GOV, its fluorescence intensity becomes brighter and brighter. So by tracking the fluorescence intensity change rate inside the GOV, we can estimate how fast the carboxylic acid can transport into the GOV. And another factors that will affect the fluorescence change rate is the flow rate inside the microfluid channel. So in our experiments, both the inside and the outside fluorescence intensity was tracked to see how the acid concentration change inside the GOV was changed according to the outside concentration. And further, to analyze this permeation process from mass transfer theory, finite difference model was developed. And the fixed second law of diffusion was used to dominate the permeation of small molecules in the bulk solution. And the membrane flux equation was used to control the membrane transfer. And in the middle of this GOV, the non-flux boundary condition was used. And the outside boundary was set according to the acid outside concentration change. And one more thing need to take into consideration is that the carboxylic acid is weak acid, so both charged species and uncharged species exist in the solution. And for the lipid membrane, only the charged species can permeate through the membrane. And finite difference model was used to solve this problem. And this is the modeling result for the acetic acid and the hexanoic acid. And we can see the permeation process of acetic acid takes much longer time than hexanoic acid. And for hexanoic acid, its internal acid concentration increased quickly as the outside concentration increased. While for the acetic acid, its internal acid concentration changes slowly after the outside concentration already reached the maximum value. And also, the concentration gradient for acetic acid mainly happened at the membrane, while for hexanoic acid, the bulk solution's concentration gradient also take a large part. So this means that compared to acetic acid, the permeation resistance in the membrane for the hexanoic acid is much smaller. And to determine the permeability for these acids, we first set a series of permeability in the finite difference model, and the internal concentration change can be obtained. And this calculated data was compared with the experimental data. and a series of chi-square values can be obtained. And as showed in this figure, and at the minimum chi-square value, it represents the best fitting of the model result to the experimental results, and then we can obtain the permeability for this carboxylic acid. And this is the result of the model fitting result. And this is for the formic acid and acetic acid. The green line represents the experimental internal acid pH change, and the red line represents the fitting result. And this is the result for the propionic acid and the butyric acid. And this is the result for the pentanoic acid and the hexanoic acid. And as we can see that as the isotyl increase, the permeability increase. And now we can summarize our result here. As the isotyl of the carboxylic acid increase, its permeability increase, and these acids oil-water partition coefficient was checked from references, and we found that as their oil-water partition coefficient increased, this is for the octanol water partition coefficient, and this is for the hexadecan water partition coefficient. And as the oil water partition coefficient increased, their permeability increased. So this means that in this kind of small molecules membrane transport, the oil water partition coefficient is still the most important factors that dominate the permeation process. And here, Overton's rule still stands for this short chain carboxylic acid. And this is my research. Here I want to thank my Professor Noah Manestad, Professor Crandall and Kim, and my lab mates for their continuous support and encouragement. And this work was supported by the NIH grants. Thank you for your attention.
There's time for a couple of questions.
Would you explain how you measure the membrane permeability on the type?
By measuring the membrane permeability, we first track the permeability, how the internal fluorescence intensity change, and then by tracking the intensity change rate, we can change this intensity to the acid concentration change, and this is the experimental data. And then we analyze this process from use the finite difference model, and then we can obtain the internal concentration change from the calculated results, from the model results. Then we compare our model results to the experimental results. As I show here, at the best fitting point, we can obtain the permeability.
One more quick question. All right, if not, let's move on.